Good morning, dear AS. We are discussing monthly test of AS Biology chapter cells, and it has 15 marks MCQs and 15 marks theory. So question 1 says, what is magnification? For this, the procedure is you'll have to find out length of this line, which is, it shows 5 micrometer. So you have to show how much is the length. We just assume that this line is say 1.5 centimeter. So this means 15 millimeter. And that also means 15,000 micrometer. And if you want, you can write on this as 15 into 10 to the power 3 micrometer. So you have the image length. So, but actual is 5 micrometer as it's given. So magnification will be 15,000 over 5. So it comes 3,000, right? It may not be same as the answer because the right answer is D, and the reason why right is D and our calculation is 3,000 because this picture is zoomed in a different level, the way I'm solving it. But the procedure, I hope you understand it. This is scale bar, and the length of this will be measured, will be converted to micrometer. That will be image length. And then that will be divided by actual. Excuse me. So the next question is about functions of microtubules, RER, Golgi body, and centriole. So dear students, Golgi microtubules make cilia and spindle. Microtubules are very small, minute, tube-like structures. So they make cilia, they make spindle, they make mitotic apparatus. And RER separates some processes from cytoplasm. Because RER is vesicles which are enclosed. So obviously, RER is like boxes, closed boxes. So there are different places inside of RER is different from what occurs in the cytoplasm. And Golgi body, vesicles are pinched off from Golgi. Some of these vesicles can act as lysosomes. And centrioles assemble the spindle during nuclear division. Centrioles have a role in nuclear division. And although their role is uncertain, but in animals, it is thought that they make spindles. A spindle means a particular shape of the microtubules. So the answer stands alpha. Number 3, as where is ATS ribosome found? So the answer is Charlie, 2 only, because chloroplast has 70S, mitochondria has 70S, and prokaryotic ribosome cytoplasm has 70S. So it's B, 2 only, Charlie. Question 4 is about enables prokaryote to identify as prokaryote. What makes you believe that organism is prokaryote? Obviously, the prokaryotes have cytoplasmic DNA. They don't have nucleus, so their DNA is found in the cytoplasm. Their cell wall is made by PPG. But the question is, having cell wall does not qualify something to be called prokaryote because plants also have cell wall. Nucleus is not there in prokaryotes. And yes, having ribosome also does not qualify something to be prokaryote because even eukaryotes have ribosomes. Prokaryotes have 70S ribosome only, and eukaryotes have 70S and 80S both. So answer to 4 is alpha, A. What is function of nucleolus? We all know, hardly anyone went wrong, but some of the students had chosen option C, which is really bad, because nucleolus does not make protein part of ribosome. Nucleolus makes rRNA. We all know that ribosome is made by 2 things. rRNA means ribosomal RNA and protein. And nucleus is the guy that makes rRNA of ribosome. I repeat, ribosome has two parts. One is ribosomal RNA and the other one is protein. So ribosomal RNA is made by nucleolus. So the answer is what? Delta. Then question 6, that is about calibration. So we have to calibrate. And dear students, you know what calibrate is. Calibration is finding the value of one graticule. And when you look at this diagram, this guy is stage micrometer. Reason because it is the the spaces seem bigger. So obviously stage micrometer will be zoomed in twice, one by eyepiece lens and the second by objective lens. So the units of stage micrometer seem far apart. So first thing you have to do is you need to identify which one is stage and which is eyepiece graticule. There are two scales, right? Okay. After this identification that what is eyepiece graticule and what is stage, I'll have to calibrate the eyepiece graticule. Calibrate means knowing the value of one graticule. So for example, there are 40 graticule equal to 0.1 of stage. Look, 1 of stage is already given, which is 0.1 millimeter. So I can write on 40 graticule equals 0.10 millimeter. Excuse me, sorry, my tablet is doing some problem. So 40 graticule equals 0.10 millimeter. Then 1 graticule should be equal to x. So we'll find out how much. So you know it's 0.1 by 40. So dear students, this comes 0.0025 millimeter, or you can call it 2.5 micrometer when you multiply millimeter with 1000. So all we know is that, we could understand 1 graticule is how much. Now this circle has the area pi r square and radius is the whole, diameter ka half. And the question is what is diameter? So the diameter is 100 graticule. You can see 0 to 100 graticule. But we know that 1 graticule is how much? 2.5 micrometer. So we should multiply this with 2.5. So diameter is 250 micrometer. But what is radius of this? Half of it. You could say 120, sorry. Radius is 125 micrometer. Now you can do what is pi r square area. So they say that a student wants to calculate, a student calculated the area of the field of view using the information there, which answer has been rounded out correctly. So, area is pi r square and pi is, 3.14 into r is 125 and then 125. I actually don't have calculated. So whatever is the answer, you have to find this way. And according to mark scheme, the answer stands delta, right? Okay, 7. 7 says, where are cisternae found? So it's 1 and 2 because endoplasmic reticulum and Golgi, they both have cisternae. So cisternae are individual sacs which Golgi body and ER are made from, right? So the answer should be 1 and 2. Let me show you what are cisternae. So look, these guys, these ones. If I could show you this, I want this image to be seen in a zoom. Yeah. So this one, These are cisternae. These are RER and every vesicle. This is flattened vesicle. Flattened vesicles, right? And then these are cisternae. These are cisternae. Inside them is the space. So we can see cisternae of Golgi as well. Both Golgi and ER have this. So look, cisternae actually is the flattened sac. These are cisternae of Golgi. You know what actually is a cisternae? Are these flattened sacs? Just one. There are 1, 2, 3, 4, 5, 6. 6 cisternae are shown here. So let's go back to what we were doing. Dear students, sun was alpha A. Now question 8 is about which row matches the function of cell structure. So this is Charlie because vacuole stores sugar and pigments. You know, pigments are the ones which give color to the plants. So chlorophyll is a pigment found in leaves and chloroplast. But there are other pigments, many pigments which are found in the vacuole. And vacuoles also store water, sugar thingy. Nucleolus makes ribosomal RNA. We have studied this. Ribosome makes protein. And lysosome contains hydrolytic enzymes. So Charlie. Question 9 is beta. Because prokaryotes and eukaryotes share only these two features. Question 10 is delta. It's again, you need to measure how long is this line and divide it by 10 micrometer. First, you'll measure X to Y and say, for example, this is 10 centimeters. You'll have to multiply this to change it to millimeter, micrometer, then divide by 10, and you'll get the magnification. Question 11 talks about which structures do not contain cristae. So it's obviously B, 1, 2, and 4 because mitochondria has the cristae. Cristae is the folds. Do not confuse cristae with cisternae. Let's see what are cristae. So cristae is something found in the mitochondria, right? And cisternae are found in ER and Golgi. So these are cristae. You can see this membrane system. This is cristae. So we are back. So question 12 is about, it's a very easy question. Which letter represents cell structures that contain mRNA? I mean, where mRNA could be found? Probably we did 11, yeah. So where could mRNA be found? The answer is beta. Because cytoplasm has mRNA. Because RNA or nucleus has mRNA and mitochondria has mRNA. Remember, mRNA is made from DNA. So if it is made from DNA, that can come out to cytoplasm and eukaryotes. But in prokaryotes, it will be already there in cytoplasm. Got it? So the answer for 12 is what? Beta. 13 question says prokaryote and mitochondria. So which row is correct for both? You know, prokaryotes have PPG, mitochondria do not have this. So this is quite obviously Charlie. Question 14 is about which statement is correct. So you all know that virus is made by a protein coat which surrounds RNA or DNA, or which surrounds nucleic acid. So the viral nucleic acid could be RNA or DNA, right? So suberin, we did not study this, but I mean, Plasmodium meta and centriole are found in plants. Centrioles are not found in plants. Suberin and cellulose are found in plants as well. No, only cellulose. And prokaryotic cells contain endoplasmic reticulum. No, they do not have any membrane bound structure. So this is A is the right answer. 15 is, which is alpha glucose. We all know that alpha and beta differ by what? Beta is up, down, up, down. I mean, in beta glucose, at number 1, look, at number 1, OH is up, at 2, OH is down, at 3, OH is up, and 4, OH is down. So it's called beta glucose. But in case it's alpha glucose, it's like down, down, up, down. So you can see the alpha and beta, how do they differ? The position of OH at number 1. So this is alpha, right? And let's move on to next question. Okay, dear students, we have to label P, Q, R, S, but the question is which process is occurring at T? The only answer for this will be exocytosis. This is theory section mark scheme, exocytosis. Should be called exocytosis things because something is being released. Second one, PQRS, name of structure and function. So what is PQRS? P is nucleolus, Q is mitochondria, R is RER, and S is Golgi body. So nucleolus, what does it do? Nucleolus makes ribosome, makes rRNA. And remember, Q is mitochondria. Many of the students, many of you had written that Q provides energy, ATP. These marks are all rejected. Because what Q does is, that tells us very clearly that mitochondria, he says, reject ATP energy, simply accept what? Aerobic respiration. It is a must. And the reason behind this is that look, in bacteria, there's no mitochondria, but still they can make ATP. So I mean, mitochondria is not a must for ATP. It is a must for aerobic respiration. So that you need to know. R was RER, and the job is what? Making a synthesis of protein and things. Majority did it right. S is Golgi body, which packages proteins. Makes the vesicles. So it's question number 2 now. Many of you lost marks in this question, unfortunately. So bacterial cell and plant cell have, bacteria and plants both have cell, but the main component on bacterial cell wall is PPG. It's PPG, which is also called murin. PPG, or you may call it murin, or you may call it peptidoglycan. Peptidoglycan, whatever you name it. And second fill in the blank was, but a bacterial cell has only small 70S ribosome. A bacterial cell does not have a large vacuole, but plant cells do have. And that vacuole is surrounded by tonoplast. Tonoplast is the membrane of vacuole. Okay, let's move on to question number 3. X, Y, and Z. What are X, Y, and Z? X is nucleus, Y is chloroplast, and Z is vacuole. X is nucleus, Y is chloroplast, and Z is vacuole. It's quite easy, I think, to identify. This big one is vacuole. These one are chloroplast 1, 2, 3, 4, 5, 6. And then this big one is nucleus. It's a very good diagram where we have nucleus. And then 1, 2, 3, 4, 5, 6, 7 are the chloroplasts. And then, so X is what? Nucleus, Y is chloroplast, and Z is vacuole. State 2 ways in which structure of animal cell differs from plant cell. Almost everyone did the right, because animals have centrioles, animals have no vacuole, plants have chloroplast, cell wall. Anyway, and define the resolution. Resolution is the ability to differentiate between two closely located objects. Your ability to see very small objects. Dear students, this is ability. Resolution of electron microscope is much higher than light microscope. You all know that electron microscope resolution is the electron microscope is resolution is 0.5 nanometer. And light microscope resolution is 200 nanometer. It means light microscope, with the help of light microscope, you can see the things if they are 200 nanometer apart. But with the help of electron microscope, you can see the things if they are 0.5 nanometer apart. So this means that electron microscope has much, much higher resolution resolving power than light microscope. So resolution is ability to differentiate between two closely located objects. This is all from my side, discussion on the whole paper. Bye-bye.